methylation arrays versus whole genome bisulfite sequencing, of course, for both of them, you have some advantages and disadvantages. So the ability to detect methylation with microarrays, of course, is limited by the probes you have on the microarray. So of course, on the microarray in general, there are probes which are of interest, but you only have this 450,000 probes on the array, which is quite good, I would say. And therefore, it is also still the preferred technology for the rapid profiling of many samples because it's easier to do, it's cheaper to do. But of course, if you're in kind of discovery mode, so if you want to detect something new, which has not been described before because you have some hypothesis maybe, then of course whole genome bisulfite could be better suited because maybe this probe is not present. The probe for your gene at this region is maybe not present on the microarray. So in this context, whole genome bisulfite sequencing might be better suited. But as I told you, it's computationally demanding and it is expensive, so you might not have the possibility. Of course, there are also some limitations in bisulfite conversion in general, as I already told you in the beginning. There could happen an incomplete conversion, so that unconverted, unmethylated cytosines are then interpreted as methylated. What also happens in this type of experiment is that there occurs DNA degradation, because due to the conditions which are necessary for performing bisulfite conversion, the DNA is single-stranded, and because of the temperature you need, and because of the addition of bisulfite, etc., DNA gets degraded very rapidly. So this is something which makes it a little bit difficult. Another problem is that I told you before that we have two different types of methylation occurring. We have cytosine methylation, and we have also the methylation via hydroxymethylcytosine. And the common technology of bisulfite conversion cannot distinguish between the two. So you're measuring the two things, but you in the end you do not know if it was hydroxymethylation or it was just 5-MC methylation. But there is also a solution for this, which is currently done all the time more, which is TET-assisted bisulfite sequencing, because this way you're able to measure hydroxymethylation separately.
What is TET?
We will see this in the next slide, I guess. It is a protein family. Here it is not written. No, I did not write it down. It's just a family of proteins which is able which is important in hydroxymethylation, and later on I think I have in some moment a slide where it shows up where I tell you how it is measured differently. But it's just a kind of protein, a special protein needed.
No.
Hydroxymethylation in general was firstly observed in 1952, but until recently, as I told you, it was not really considered important. But it was recently found that there is a high abundance in embryonic stem cells that is highly tissue specific, and that is important in human and mouse brain cells, so it might be good to investigate hydroxymethylation as well, because there's now a lot of evidence that hydroxymethylcytosine also plays an important role. And what we also know is that the amount of hydroxymethylcytosine seems to increase with age, for example. But as this is a very new topic, the functions of hydroxymethylation are not yet fully discovered. What is thought that it is maybe an intermediate step in active DNA demethylation. So as I told you some time ago, in the early embryonic development there is occurring this general demethylation of everything, and it is thought that hydroxymethylation plays an important role in this process. It also may have important function in general as an epigenetic mark, what we do not yet know. In embryonic stem cells, it is associated with the self-renewal capacities of these cells. It seems to be especially important, as I told you in the previous slide, in the brain. So in central nervous system, there is a lot of hydroxymethylation being found. It has been associated with neurologic diseases and also with acute myeloid leukemia. So it seems to be an important epigenetic modification as well. Here we have again the TET-assisted B-sulfite sequencing, although it's not written down here what it is exactly, I'm sorry. TET. Yeah, for sure it's an abbreviation for something, but I do not know right now what it is, and we can look it up later maybe. I do not work with hydroxymethylation, but I think it's important to mention that it is another type of methylation which we are currently discovering. So, what you do when you do TET-assisted B-sulfite sequencing, where you include these TET proteins, is that the 5-HMCs in the hydroxymethylation gets protected by vacuumization, which is followed by MTET1 mediated oxidation, which is called. And then you do again a B-sulfite treatment. And here in the end, the hydroxymethylated C reads again as a C, and the methylated C reads in the end as a T. So then comparing again with the original sequence without having modified anything, you can then know, you will then know if it was hydroxymethylated when you have a C there, or if it was not. So, for the last part of the presentation about DNA methylation, I want to tell you a little bit about a study I was participating in, which we published last year, where we worked on DNA methylation as well. The publication is entitled Epigenomic Analysis Detects Widespread Gene-Body DNA methylation, hypomethylation in chronic lymphocytic leukemia. So what we showed here is that it is not, as previously thought, that only the methylation in the promoters is important, but we also have evidence here that gene-body methylation plays an important role in the development of leukemia. And I will present you a little bit about the steps we did in this analysis and why it was important in this context to look at methylation profiles. Just a few words about chronic lymphocytic leukemia, which is the cancer I'm working on. Chronic lymphocytic leukemia is a blood cancer which affects the B cells, so cells important in our immune system, which has two important clinical and molecular subtypes. So there is the subgroup which is called MCLL and the subgroup which is called UCLL. In MCLL you have a region which has a high rate of mutations, which is the immunoglobulin covalent gene region. It is not so important to understand what this is exactly, but what is important here is that MCLLs, so the mutated CLLs, generally have a good clinical prognosis. So these patients, normally they do not require treatment. They have the disease, but they can just continue with their lives. And in general, they do not have a high risk of dying from the disease and of requiring treatment. So they have quite a good prognosis. But in the other type, they have worse prognosis. So they reach faster to a state which needs treatment and which could lead to death finally. So what we wanted to know in this study is, as we know that there are two different subtypes, what would be the normal counterparts of these leukemic cells? So we know that there are B cells, but we have several different B cells in our blood circulating.
Oh yeah, no, that would be like the difference between the two subtypes. It would be like the proportion of healthy cells, or what would be?
Well,
or how can a patient be okay still having leukemia.
No, he's not okay, of course, but he has a lot of better prognosis and he does not even require treatment. So it's a chronic disease, which is when you have the first subtype, although you have leukemia, you have symptoms maybe, but you're not in such a severe state that rapidly as compared to the other group. So it's a chronic disease progressing at a very slow pace. And as you get leukemia at an advanced age normally, so you are maybe already 60 years old or something, and for 20, 25 years you probably can live without treatment, so you're reaching already an age in which maybe you will die from other reasons as well. So it is not considered as severe as the other type of leukemia. Of course, it's always severe to suffer leukemia. But if you do not need treatment, it's another thing. Because you can just continue with your life and you have your control, etc. But it's quite different, the two types. And we wanted to know what is the normal cell counterpart of these subtypes, because for some reasons there must be these differences. So what we had available for this study were the DNA methylomes of altogether 139 CLL patients. So you see again, that is quite nice for statistics if you have a lot of samples available. So we had a good number of patients right here with the, but for whole genome bisulfite sequencing, we just did for each subtype one of them. For the other ones, we used the same methylation area I presented you, where we had about 60 UCLLs, 76 MCLLs, and 6 with undefined mutational status, because you cannot always identify exactly if it is the one subtype or the other. And we also had normal counterparts of normal B cells, where we also did whole genome bisulfite sequencing and methylation array for several subtypes of memory B cells. You see that we had several different steps in the cell differentiation of B cells available right here. So what we did first of all was that we compared what happens in the normal differentiation process of B cells. So if you start from naive B cells until non-class switch B cells and memory B cells and class switch memory B cells, not so important what's the difference between them. It's just the naive is the most immature one, and then it differentiates and gets more mature. And you can see that with every step, the methylation seems to get less. So there occurs a hypomethylation. Here again, you have the values. So the darker the color, the higher the methylation. And you can see that the unmature cells seem to have a lot more methylation. This is whole genome, so you have all the genes right here for all the chromosomes, so chromosome 1, 2, 3 until 22. Mm-hmm. And you see that when you compare it to the next step, here you have already lighter blue, and then it gets even less methylated during differentiation. You can also see this here in the figure on the right, where you have here the later differentiation step, and here the more naive B cell, and you can see that the methylation amounts are higher. So what we then saw that is that the two subtypes I told you, so UCLL and FCLL, when you look at their methylation profiles, they seem to be quite different. So here, the red dots are MCLLs, and the yellow or orange dots are UCLLs. The other ones are the healthy counterparts we had available. So here we have naive cells, and the memory B cells, the more mature ones. And what you can see is that when you look at their methylation, when you do this kind of analysis, they separate quite well from each other. There are some outliers here, but you have one group here and one group here. While when you're looking at gene expression, you do not really see this pattern.
Mm-hmm. By which method are you assessing the gene expression?
This is also with microarrays done. So here we have the microarray methylation, and here we have gene expression microarrays. And the kind of analysis, what have been done here is just a normal principal component analysis, where you can see then how close to each other are your different samples, or how far from each other. So this is just another example of what I told you before, why it could make sense to look at methylation instead of gene expression. Because with methylation you see already some difference between the subtypes, but with gene expression it is not so clear. Also, I have to admit, when you do this analysis with gene expression data and you look at principal component 2 and 3 instead of principal component 1 and 2, you already see some kind of separation of the groups. But again, it's not so clear as when looking at the methylation profiles. So what we then did is that we did a hierarchical clustering of all the samples together. And what resulted from this analysis is that UCLL seems to be epigenetically more similar to naive B cells, and MCLL seems to be similar to memory B cells. So you have this cluster of memory B cells here together, more or less, with MCLL, and vice versa for the other subtype. So what was proposed then is that probably the naive B cell is the one where UCLL is originating from, and MCLL probably derives from the memory B cell, as epigenetically they seem to be more similar to each other. So therefore, we then compared the different CLL subtypes to their normal counterpart we saw before in the hierarchical clustering, and compared again their methylation patterns. And what we saw is that there occurs again a lot of demethylation when comparing the healthy cell with the leukemic cell. So again, you have the same representation of the colors in the figure right here. And there is a lot of hypomethylation going on there. What was very interesting then is when we did a consensus clustering of the methylation profiles, we identified 3 CLL groups. So classically, we know that there are these 2 molecular subtypes, UCLL, MCLL, separated from each other. But when we did this unsupervised clustering, we had an intermediate group. And interestingly, this intermediate group, which we saw here, also seems to have a kind of intermediate hypersomatic mutation, because this is the color scheme of the IgHV somatic mutations, which is this region which is used to identify the different subgroups. And they seem to be in kind of intermediate values here as well. And what was important here is that when we did a kind of analysis for time to treatment for these patients, so the time of progression of disease until they needed treatment was also intermediate for this newly identified intermediate group. So we have the NBC-like CLLs with bad prognosis, we have the MBC-like CLLs with good prognosis, and this new group, which was previously unknown, also lies in between in this context. So it was really a good thing to look at DNA methylation right here, because we could identify a kind of new clinical subgroup, which might also be something which later on could be used then to find new therapeutic targets, looking at the things which are happening here in methylation originating from the different cell types. So I guess this is already the end of the methylation part. So, yeah, I already took a little bit more of time we had available, I guess. But if you have some questions still, and if you do not want to go home already, I am here, so.
Well,
Any
you
questions?
first.
No.
It wasn't, I mean, it's a technical question about the process for the typical analysis.
Yeah.
In which you said that you separate into batches of different nucleotides.
Yes.
And then how do you put together again in the sequence the whole thing? I don't understand it. Or what do you mean by putting separate batches?
I will try to find an example. So maybe you have a sequence like this. And then what you do is you have to fragment and amplify the DNA. And when you do this, you add synthetic bases to your batches, which then impede the elongation of the transcript. So in the end, you have them in one batch. So probably you have a blue batch, a yellow batch, a red batch, and a green batch. And for every base, you have your certain color. So what you're doing is here, you add the modified T, for example, and you will end up with one sequence which is T, and another one which is TAT. And the T, in the end, is labeled blue. Then probably for the A's, you used the yellow. So you have then in the end a sequence which ends in the A. The A is the one which is marked and where it cannot grow longer anymore. So it stops with the A. The next step is then the same for the red one, which would probably be a C and a G. So it ends here with the red mark. And in the green batch, you have this one. And then what you do is when you put it to your sequencer, they will run according to the length. So the output is then, first of all, you have a T. The next thing you will read is a TA. Then you will have TAT, etc., etc.
It's like ordered.
It's like ordered, and
Yeah.
as you have labeled them with different colors, your scanner here just reads blue, and here it reads yellow. And this way you have the colors, and as every color is corresponding to a base, this way you get the sequence in the end, as it comes out as a color measurement of intensity from the scanner. I don't know if it's clear.
No, it's the previous thing now is clear. Now I got the doubt of like the intermediate ones, but don't worry.
Yeah, I know it's difficult the first time you hear it, but I can try to explain it if you. What's your new doubt?
No, I mean, just like in the sequencer, if it just reads like pieces of different colors, but
Yes.
you have some other nucleotides in the middle, which are well.
Yeah, but as you have them ordered, in the end, you know what you're reading, no? Because you know that here you had the T. So first you start with the T, then you had the A, and then you had again a T. So you're getting your sequence out of it again. Because as they come out ordered, because of the different weights, because of the different numbers,
I see.
you can just you have them reordered already. But what is complicated indeed is that you cannot create DNA frequency of an eternal length. So you have in the end reads of as I told you before, maybe 200 bases or 500 bases or whatever. And what we then have to do with bioinformatic approaches is to find the regions where these reads are located in the genome. And as there are repetitions of sequences, etc., this is a difficult task because you do not always know where it really lies in the genome. So this is the really difficult step. But the short sequences just come out like this.
Yeah, all right. Thanks.
Yeah. A ustedes. Venir un viernes por la tarde tan tarde. Muchas gracias. Es el hotel se llama Harrison, pero está muy cerca de aquí.
Está en el periférico.
Periférico del sur, ¿verdad? Hay un centro comercial que está directamente al lado.
Sí, periférico.
Ah, vale, sí, se llama así.